In this lecture, we will talk about secretion of certain substances by the proximal tubule. Is the urine formation process is going on. The first step in the urine formation is filtration. The second step is tubular reabsorption, and finally there is secretion. Not all the substances, not all the solutes are secreted by the tubule, but there are certain substances, especially the metabolic waste, which are secreted by the tubule, especially the proximal tubule. As we have discussed in detail the different steps of urine formation, the filtration process, the tubular reabsorption, and then we have discussed the mechanisms through which reabsorption of glucose, bicarbonate, sodium chloride, potassium, etc. occurs. Now we need to discuss the secretion process, because we have discussed in detail the proximal tubule and the importance of proximal tubule in reabsorption. So we also need to know about the importance of proximal tubule in secretion, because overall all these steps are contributing to the urine formation process. Now the secretion process is simple. Is the tubule is reabsorbing certain substances like glucose. For example, 180 gram of glucose is filtered here per day, and out of this 180 grams is reabsorbed into the blood, into the peritubular capillaries, and 0 glucose is going into the urine. So the reabsorption process of the glucose is very active. Similarly, the reabsorption of bicarb, sodium and chloride are also very active, although not like glucose, because some amount of these bicarb, sodium chloride are going into the urine, but still they are actively reabsorbed. Similarly, there are certain substances like bile, oxalate, urate, some drugs and compound PAH. They are secreted. Now the secretion is basically the reverse of reabsorption. The reabsorption is basically the taking in of these substances from this lumen into the peritubular capillaries. Now if these cells are basically cells of the proximal tubule, this glucose amino acid, they are reabsorbed into the cell and taken into the blood vessel. But in secretion process, these substances will enter from the blood into this intercellular spaces, and then they will come all along the tubule and they will be secreted here. And the purpose is that the body wants to get rid of these substances, and these are basically thrown out in the urine. Now the main important substances which are secreted, they include the metabolic byproduct like bile, oxalate and urate. Bile, oxalate and urate, these are secreted into the tubular lumen. Apart from these metabolic waste, there are some breakdown products of certain hormones. Then some drugs, for example, the penicillin and salicylates. Those drugs are also secreted into the tubular lumen, and secretion is the reverse of reabsorption. And secretion basically increases the speed with which the body wants to get rid of the waste material. Now, the body also secretes the drugs like penicillin and salicylates. Now, this thing creates a problem because the body gets rid of it, and the level of penicillin and salicylate in the blood basically drops because of active secretion. So it creates a therapeutic challenge as well, because the level of the certain drugs has to be maintained steady in the blood so that the drug can act properly. For example, penicillin level in the blood has to be maintained so that it can act on the bacteria and kill the bacteria. If it is actively secreted from the peritubular capillaries into the tubular lumen by the proximal tubule, then its level in the blood will keep on decreasing. So multiple doses are taken every 6 hours or 12 hours, or like for every drug there is certain dosage in which they are taken. Not all the drugs are secreted in this way, but certain drugs. Now similarly, just like penicillin, salicylates are also secreted in this way. Apart from these waste products, metabolic waste products and drugs, a compound known as para-aminohippuric acid, PAH, is also very actively filtered and secreted into the tubular lumen, and the body wants to get rid of this compound very quickly. Now this thing is very helpful, because whenever the doctors, the physicians, the scientists, they want to check the functions of the nephrons and the functions of the kidney, they can use this PAH to see the clearance, the rate of clearance by the kidney. So the secretion can create a therapeutic challenge when they secrete the drugs, but secretion can be basically helpful when you look at the secretion of the para-aminohippuric acid. Similarly, secretion is also helpful because through secretion the body basically quickly get rid of certain waste like bile, oxalate, urate. So the secretion is basically, you can say, the final step of urine formation in the proximal tubule. After the proximal tubule, the filtrate will then go into the loop of Henle and the distal tubule, and then medullary collecting ducts. But when in the proximal tubule, the filtration has occurred, reabsorption has occurred, and the secretion process has occurred, the urine formation process for the proximal tubule ends. These are the steps in the proximal tubule. After that, the filtrate will move on, and the different parts, the loop of Henle, the distal tubule, will be doing their own actions. But the secretion ends the function of proximal tubule in the urine formation. Now, if you see, This graph we have discussed in detail in our last lecture, and we discussed that along the proximal tubule, some substances are reabsorbed, some substances are not reabsorbed. So the concentration of certain substances keeps on decreasing along the length of proximal tubule, like glucose and amino acid. They are very actively reabsorbed, and they are taken back into the blood, so their concentration in the tubule keeps on decreasing. But the creatinine is not at all reabsorbed, so its concentration in the tubule keeps on increasing. Similarly, those substances which are secreted here, which are secreted into the tubule, their concentration keeps on increasing in the tubule because the body wants to get rid of those substances. So that's the end of urine formation process by the proximal tubule. After that, we will talk about the steps of urine formation in the loop of Henle. Thanks a lot for watching the video.